Hello, we are student of Bachillerato from Zaidín Vergeles, and I'm going to explain the introduction to this project about pepper. The pepper is a voice plant that can be found in different colors, choose a red, yellow, orange, and other color when the fruit ripens. Due to her high water content, they are low in calorie and are good to antioxidants, choose vitamin C and A. The great versatility of the pepper fruit and the considerable increase in production mean that it is great interesting and great economy important.
Spain is one of the most important countries in the world in pepper production and consumption. The pepper can be stored at room temperature in the fridge or frozen. However, it is unknown under what condition it shall be stored to maintain its organoleptic, nutritional, or healthy properties. In this sense, the main objective of the research is to study the variation of some properties and nutritional requirements of pepper over time in relation to the way in which we store vegetables.
Now we are going to read the objective. First one. To study change in organoleptic physics in pepper fruit, stored under different temperatures and time conditions. Second one, to ask the influence of strontium condition on the deshydratation of pepper fruit.
Third one, to follow the time course of sustained biomolecules like solute sugar or proteins in pepper under different temperature conditions of store. Fourth one, to compare change throughout time in ascorbate vitamin C and carotenoids source of vitamin A in pepper fruit suggested to diverse store conditions. And 5, to assess the best condition of pepper store in order to preserve its organoleptic and nutritional features.
Now we're going to explain the materials and methods that we use. First, our plant material was pepper, specific the Capsium annulum L, which is also called Dulce Italiano from a local market in an immature green ripening stage. Here we were working with 10 different peppers that we divided in 3 groups depending on the way of strength. We have the room temperature, fridge, and freezer. This is a general scheme of the experimental design. Here we have our what we were analyzing, for example, the fresh weight or bricks and pH, crude extracts, vitamin C, protein, and also pigments. We were taking measures the first day, the day 0, and in a week, in 2 weeks, and in 4 weeks. These are the methods that we were using. First, we observe and touch the peppers in order to study the organoleptic features. Also, we measure the fresh weight and the dry weight, and the soluble sugars in a refractometer by bricks. Also, the pH and buffered and ethanolic extracts that the first of them, the buffered extracts, were used in order to determine the vitamin C using also Lugal, and the protein determination by Bradford method. And the ethanolic extracts were used in order to determine the photosynthetic pigments.
Okay, so we start with changes in organoleptic features. The pepper stored at room temperature started ripening from week 1, and after 4 weeks, they were fully red. While the peppers from the fridge and the freezer kept their green color. Another thing we observed was that the peppers that were frozen became flaccid because their cellular structure was broken due to increase of water volume. about water loss, every pepper loses water regardless of the preservation method used. But we observed that the peppers stored at room temperature had a 12% of water loss. The ones from the fridge had a 3% of water loss, and the ones from the freezer 2%. And in this other graph, we can see that there is no noticeable change in dry weight of the peppers.
Well, the Brix is the percent of soluble sugars in the pepper, and it was measured by means of a refractometer. The graphic shows a decrease of the Brix in all the temperature environments. Fridge and freezer ends up with a barely difference in the final results, but at room temperature, the degradation of the sugar is smaller. This may be the result of a water loss that brings a higher concentration of the sugars in the pepper. In the graphic of the pH remains between 6 and 4.5, where the most remarkable fall was at room temperature, from 5.5 to 4.5.
So now talking about the results of the vitamin C, we could conclude, as we thought at the beginning of the project, there is a decrease in the value of the vitamin C throughout time. The amount of vitamin C decreases in the first 2 weeks, but if we look at the graph, we can notice that the freezer peppers lost more vitamin than those that were preserved at room temperature. Moreover, from the second week, we observed that the results of the vitamin C in the fridge and freezer peppers were mostly constant. But nevertheless, there was a rise in the results of vitamin C that were conserved at room temperature due to the water loss.
In the case of proteins, we can observe in the graphic that there aren't any significant change. Actually, if we ignore the first point, the rest of them are similar, but there are some little difference depending on the storage location, which is actually uncommon because depending on several articles, proteins usually don't vary much depending on the storage location. So our hypothesis is that this variation is caused by the water loss. In the case of the freezer, for example, when the pepper freezes, the cellular structure breaks, releasing water and causing water loss.
So now we are studying the pigment concentration of chlorophyll A and B and also carotenoids. We have 3 different graphs depending on the temperature condition of storage. The first one is at room temperature, that we can see how chlorophylls A and B decrease and carotenoids increases. That's because of the ripening process. Here between the day 7 and 14, there is a big variation. And then in the last day of measurement, the 28, we can see how there isn't any carotenoids, there isn't any chlorophylls and many carotenoids because of the ripening process was at the end. And this graph is at the fridge. Here we can see how there's a general degradation of all the pigments. That's because there isn't like a ripening process, it's only degradation. Here in the freezer, we can see an important change, and that's why when we were studying the samples, were in the fridge, in the freezer, because there wasn't any important change, and that's why we can study them easily.
So finally, these are our conclusions. The first one is that ripening symptoms color shift from green to red were only observed in fruits maintained at room temperature, and this was confirmed by the profile of the carotenoids content. The second one, independently from the time of temperature incubation conditions, the pH in fruits ranged to 4.5 to 6, and there was an overall decrease in the content of soluble sugars throughout time. The third one, vitamin C content mostly lowered with time in all temperature treatments. The next one, the major changes in the content of pigments were observed in fruits maintained at room temperature. And the last one, a global assessment of data in the conditions applied in this study indicates that the incubation at room temperature seems to be the most appropriate practice to maintain the best organoleptic and nutritional properties of pepper fruits.
So that's all. Thanks for your attention. Here we have a photograph of the rest of our teammates that couldn't come here today. And also we want to thank especially our biology teacher, Antonio Quesada, and Jose Palma, and also Marichu for giving us all the help, and in general the opportunity that was gave to us. So if you have any question, here we are. Thank you.
Se abre el tiempo temido por los estudiantes, el tiempo de las preguntas. ¿Quién tiene alguna?
En primer lugar, daros la enhorabuena a todos porque habéis hecho un trabajo increíble, aparte de presentado desde que en la pasada. Entonces me ha surgido una duda de la última diapositiva en la que podéis poner la de los pigmentos. No sé si lo habéis contado y no lo he seguido, pero ¿por qué habéis podido averiguar por qué en el congelador hay este pico hacia arriba después de la semana? ¿Por qué aumentan todos los pigmentos a la semana? Luego ya disminuyen, pero...
A ver, tenemos que tener en cuenta que esto es un estudio preliminar que hemos hecho. Hemos utilizado solamente 10 pimientos. Deberíamos realizar un segundo proyecto donde utilizáramos unos 100 para poder calcular medias y variaciones, porque dependiendo de cada pimiento como individuo, pues tendrá unas características que pueden variar un poco más. Pero igual, si nos damos cuenta, la curvatura, el pico que hace es igual en todos los pigmentos, y en muchos artículos que hemos nosotros leído de biografía antes de realizar el proyecto hemos visto que la variación en el congelador de los pigmentos es nula. Entonces, pues por eso hemos visto que la gráfica tendría sentido de ese modo. Espero que haya respondido a su pregunta.
A mí, en relación a estos gráficos, se me plantea una pregunta, digamos, que quizá tiene que ver de muchos de los gráficos. Hemos visto diferencias a mantener los pimientos a temperatura ambiente frente a los dos que están en nevera. Efectivamente, una de las diferencias importantes es la pérdida de agua. Entonces, cuando hacéis referencia de una cantidad de un determinado metabolito o lo que sea frente a gramos en cada muestra, cuenta el agua que había en esa muestra. ¿Y no hubiera sido más correcto hacer la presentación frente a gramos seco de la muestra? Es decir, haber quitado el agua de todas las muestras. No sé si me estoy explicando. O sea, lo que quiero decir es que si el agua genera un problema en la referencia, habría que quitarla. O sea, si realmente queremos saber que el pigmento no se incrementa o se pierde, tendríamos que igualarlo. Pregunto, no lo sé.
Sí, pero nosotros cuando solemos consumir los pimientos, mayoritariamente los consumimos con ese agua. Entonces queremos estudiar el cambio que se produce, incluyendo toda la parte del pimiento, también el agua.
No, eso es correcto. Lo que yo me estoy preguntando es, por ejemplo, vete a gráficas anteriores. Quizá la primera incluso. Bueno, esta,
Esta, la de la pérdida.
La del agua es correcto. Vete a la siguiente.
Esta.
No, una más. Esta, por ejemplo. O sea, estamos midiendo el contenido en vitamina C. Y bueno, podríamos pensar, se mantiene más o menos la cantidad de vitamina C equivalente, o lo que resulta raro es que a temperatura ambiente haya más vitamina C tras 28 días que en la nevera. No sé, o sea, resulta raro. Yo no digo que no sea verdad. Y entonces me he preguntado de si el hecho de haber referenciado frente a gramo de pimiento no está poniendo un error. Y tendría que haber, no lo sé, pregunto.
A nosotros también nos ha parecido curioso que a temperatura ambiente sea donde mejor se llega a conservar la vitamina C en el pimiento.
Pero bueno, también os digo, puede ser, ¿eh? O sea, pero bueno. ¿Alguna pregunta más? Es que yo soy muy follonero, perdonad. Ahí al fondo, a la izquierda.
Teniendo en cuenta este experimento, ¿cuántos serían los días más o menos que hay que esperar para tener el mejor equilibrio de vitaminas C y proteínas para el propio pigmento?
Yo creo que estudiando la vitamina C, nosotros lo hemos hecho en un periodo de más o menos un mes, por tal de tener un patrón de semanas en las que medir y obtener resultados concretos. Pero si tu pregunta es en qué punto alcanzaría mayor vitamina C a temperatura ambiente, por ejemplo, creo que no podríamos determinarlo así a simple vista, porque bueno, tendríamos que hacer otro estudio más, ese podría ser segunda parte del estudio, o lo que realmente sabemos es que a temperatura ambiente siempre vamos a tener más vitamina C que pimientos congelados o que pimientos refrigerados. No sé si te he respondido más o menos.
La pregunta era, ¿cuándo nos tenemos que comer esos pimientos?
Bueno, se acaban...
Iba a puntuarte que,
Perdón.
para lo que ves en la gráfica, si te, preferiblemente sería en el día cero, que es cuando más vitamina C hay, pero...
Bueno, pues ya damos un poco por finalizada la presentación de los distintos proyectos, que no del Congreso, y permitidme que haga una pequeña presentación, bueno, un aplauso, por supuesto.